Good evening, everyone. Today is my week 8 session. So, in this session, we'll be talking about the problems related to week 2. So, in week 2, you have covered Human Protein Atlas, affinity based proteomics, clinical considerations for omics, and Integrative Genome Viewer. So, along with this, we'll practice some MCQs and we'll also look into the current literature that what is currently going. And I'll also be taking your doubts related to the assignments of this week 8. So firstly, we'll talk about Human Protein Atlas. So in this session, what I'm going to do is I'm talking about Human Protein Atlas in complete detail because in the lecture, many of the things were already covered. So I thought that instead of repeating them again, I'll just talk about new things. So let's explore Human Protein Atlas. So Human Protein Atlas is like a comprehensive resource which maps the expression and distribution of proteins related to human tissues and cells. So it will contain all the information of proteins which are expressed and distributed across tissues. And it was launched in 2003 and it contains the data which is coming from the experiments such as antibody-based imaging, transcriptomics, and proteomics. And it will tell you the information such as where the protein is located, what is the abundance of the protein, and what is the role of that particular protein in the human body. So in the session when they were talking, so they mentioned that it was recorded in 2018. So till that time, it was not much progressed. But as current in the current time, now there are 12 different sections. So previously there were only three main sections of Human Protein Atlas, which was Cell Atlas, Tissue Atlas, and Pathology Atlas. But now there are more and more sections which have been added to it. So we'll look into them one by one. So those 12 sections are the tissue section, brain section, single cell type section, tissue cell type section, pathology section, disease blood atlas, immune cell, blood protein, subcellular, cell line, structure, interactions. So now, with the advancement of technologies, more and more data has been collected. So they have figured out and they have divided that data into 12 different sections. So when we talk about tissue section, it means that it will be talking about the distribution of proteins across all the major tissues and organs in the body. So whenever the samples were collected from different tissues, then proteins might have been identified. So that on the basis of that, they have divided that which particular protein is expressed in which tissue. So whenever you will go to this section, it will tell you tissue-wise protein expression and other information such as its functioning and what are the antibodies which are complementary to that particular protein. Then in the brain section, it will be entirely dedicated to brain. So there are multiple regions of the brain. So the proteins are distributed variably in different regions. So it will tell you about that information, and mostly it is about the mammalian brain. But uh, till now, they have added two other information as well about the mouse brain and the pig brain. So now with more and more advancement, they might add some other organisms as well. But currently, it is dedicated to the mammalian brain, which involves human, mouse, and pig. Then comes single cell type. So with the advancement of single cell RNA-seq and other techniques which are focusing on single cell, so the information related to single cell also can be gathered. So it tells us the expression of protein coding genes in human cell types, which is based on single cell RNA-seq. Then tissue cell type. So in this also, it will tell you the information of the tissues where that particular protein is expressed, and in that tissue, what compartment of the cell it is being expressed. In the pathology section, it will tell you that what is the impact of that particular protein, because we know that there are some genes or proteins which are linked to some disease, such as cancer or any other disease. So in pathology section, you will be able to find that also, that whether this particular protein is involved in any kind of disease or not. Then disease blood atlas. So in this, the patient's blood is collected as a sample, and from that, we get the proteins. So in that way, it will tell about the disease, the proteins which are associated to disease and which are present in the blood. Then immune cell. So it tells about the protein coding gene expression in immune cells such as T cells, B cells. Then blood protein, it will tell about the proteins which are expressed in the blood and which are secreted by human tissues in the blood. So not exactly about the disease, but normally which proteins are present in the blood. Then subcellular section comes. So it means that in the subcellular localization of a protein will be discussed. That inside the cell where it is expressed, whether it is expressed in the nucleus or membrane or Golgi body or mitochondria, which organelle. So that all things will come under subcellular section. Then comes cell line section. So it will tell the expression of protein coding genes in human cell lines. So mostly when we say cell line, we talk about the in different cancers when we collect those cell lines which are growing rapidly, so that are referred to as cell lines. So mostly dedicated to cancer. Then comes structure section. So in this, all the proteins which are identified by all the experiments, their structure is given. So if you want to find the three-dimensional structure of any protein, we can go to structure section. Then in the interaction section, we'll explore that uh, what kind of other proteins a given protein interacts with. So all these sections comes under Human Protein Atlas. And now we'll see, yeah. So if we talk about the advancement, that how this Human Protein Atlas has grown. So these are the years and these are the features which added on with advancement. So firstly, it was launched in around 2003, and in 2005, there are 700 antibodies which were present. So just give me a moment. Yeah, okay. Yeah, so in 2006, there was inclusion of cell line data and confocal images, which shows subcellular localization. And then in 2007, the new search function came up, which allowed advanced query-based searches was included. So you can go through this and you'll find that how with each year new information has been added to Protein Atlas. And this is also another page which shows that how different additions have been done. So firstly, in 2015, it was about the tissue-specific information. Then in 2017, subcellular information was incorporated into Human Protein Atlas. In 2017 only, cancer information was also incorporated when they were able to identify that which proteins are specific to which particular cancer types. For example, BRCA is a protein which is mostly overexpressed in the breast cancer. So in that way, proteins can also be categorized on the basis of cancer, although there are multiple proteins which are combinedly expressed in all types of cancer, but still there are some proteins which are specific to some cancers. So in that way, we can target those proteins. Then comes blood protein atlas. So it came in 2019. And at that time, when these lectures were recorded, that time they were about to add this blood part. And now in 2000, means after 2019 and 2020, they also added the brain part in which the the proteins which are expressed in brain, they were also added. So yeah, it tells about the milestone which they have achieved through these years. Yeah, so now let's begin. So this is the home page. This is a small section so that you can see it clearly. So whenever you go to the Human Protein Atlas, you can follow this link and you will be taken to this particular page. So there will be 12 sections which will be mentioned in the left side. So when you click on this section option, these all sections will appear up, in which the first option is the tissue. So when you click on that tissue section, it will elaborate that what does this tissue section contains, and with so as new data is updated, they will also give you some examples of the just latest proteins which have been identified in a tissue. So in this, they are giving the examples, and in this, they are also telling you that which kind of evidence is available for the proteins. So the evidence is available at mRNA and protein level, which means that whenever different experiments have been done, as you know, that in RNA-seq, we only get the information at the level of RNA, that this particular RNA is expressed. So we already know human genome, and from human genome, we also know we can also identify the human transcriptome, that what RNAs could be synthesized. So in that way, in RNA-seq, we can validate that this particular RNA is present, but not all RNA will convert to protein. Means in here RNA means mRNA, because there are other RNA also which do not code for protein. But if we talk about mRNAs, then not all mRNAs convert to protein. So in that way, from RNA-seq, we only get the evidence at the level of mRNA, and we don't know whether it will be expressed or in which quantity it will be expressed. So after that, we need to perform some protein assays or mass spectrometry to get the evidence at the level of protein. So similarly, in tissue section, we'll be having the information related to the proteins both at the mRNA and protein level. So when we'll explore it, we'll see that it will tell us that information also, that at what level evidence is present. And this particular tissue section will contain the information of 44 normal human tissues, and which was derived from antibody-based protein profiling and using the immunohistochemistry. So in antibody, the immunohistochemistry is used. So, and additionally to this, the other protein-based assays are also used. So in this, they are only telling about it. And they've also mentioned that how many genes are present. So the protein data, it covers around 15,303 genes. So currently, human genome has around 20 to 22,000 of genes, out of which these genes have been identified by the Human Proteome Atlas, for which the antibodies were available. So as we know that to identify a particular protein, we need something that how we will capture that information. So in this section, they are using the immunohistochemistry. So for immunohistochemistry, there are some antibodies which will bind to my protein of interest. So if some protein is present for which antibody information is not known, so that in that case, I cannot identify that protein. So that's why only 15,303 gene information is available. And for other genes, we do not have the respective antibodies. That's why they were not identified. It doesn't mean that those genes are not present. Those also be present, but for them, we cannot identify them. And then in this, it will tell you the protein localization in tissues at the single cell level. And if a gene is enriched in a particular tissue, so it will also tell you the enrichment, that whether it is specific to that particular tissue and what is the expression level, and which genes have similar expression profile across tissues. So all these informations can be captured from the tissue section. And if we scroll further, then we'll find the tissue and organ proteome. So in this slide, so in this particular, when you scroll down, so in this particular image, it tells about all the tissues about which the information is available. For example, in eye, the retina, from where the protein has been identified. So this means tissue enriched, group enriched, tissue enhanced. So it is categorizing those proteins which or genes which have been identified in these particular tissues into multiple sections. So red color means these are the tissue enriched genes. That means these are enriched in this particular tissue. Then comes group enriched. So there are some group of genes which are together enriched in a particular tissue. So this particular will tell us that how many number of group enriched genes are present, and then tissue enhanced. So there are certain genes which are overexpressed in one particular tissue as compared to other. So in that case, those will come under the tissue enhanced genes. And then there are low tissue specificity genes, which means that those genes can also be present in other tissues as well. So they will be low tissue specificity. That means they're not specific to that particular tissue. And then there will be certain genes which are not detected. So that also it has been mentioned here. So similarly, for all the tissues, you will find the information. For example, if we want to explore that which genes are present in brain, so we can simply click on this brain option. So if you do not click and if you just keep your cursor on brain, then it will tell you that how many brain elevated genes are present. So out of them, the enriched genes are 475, group enriched genes are 457, and enhanced genes are 1265. And if you want to get more information that what are these genes, so you can click on it and they will redirect you to those genes. But now, as of now, we are not clicking on that. We'll see it in the later part. Now, if you further scroll down, then it will tell you that how the genes are distributed. So means in the at the level of tissue specific data set. So if we talk about the specificity, so the tissue enriched genes were about 3,150. So in the previously, as we have seen that there were total around 15,000 gene information which was available. So from that, 3,150 were tissue enriched, which was specific to some tissues, and then 1,583 were group enriched, then 6,197 were tissue enhanced, and then 8,194 was low tissue specificity, and 1,038 were not detected. And we talk about the distribution. So this 1,125 were detected in single tissue, which means that they are only present in one of the tissue. Detected in some, which means that they might be present in more than one tissue, but not in all. Then detected in many, which means many tissues it is present. Detected in all, which means there are certain genes which are expressed in all the tissues, and not detected, for which the antibody information is not available. We could not detect them by using immunohistochemistry. And then if you further scroll down on the same tissue section, then you will find they will provide you the network of the enriched genes. So in this, for each tissue, they have provided the list of enriched genes as well as the genes. Uh, just give me a moment. So yeah, so group enriched and tissue enriched genes. So this red color represents the tissue enriched genes, and this orange color or golden color represents the uh, group enriched genes. So for each particular tissue, the links are provided. And those group enriched genes can be shared among more than one tissues. So that's why this network was constructed. And you can simply click on any of these tissues and it will take you to the information of genes, all the genes which are expressed in this particular tissue. So why we are saying gene and not protein? Because there will be some genes for which the evidence might be available at RNA level only. So that's why we are referring to them as genes. But it will contain the information about the proteomes or the proteins. So don't get confused with that. Yeah, so if we click on this heart, so it will take us to the another window in which all the gene information will be given. So if you are interested in the heart muscles, then it will tell you about all the genes which are expressed. So this is just a screenshot, but if you will click on it, then another window will appear in which all the genes which are present in that heart muscles will be shown to you. So there are total 138 genes which were found. So what information you can find? So it will firstly tell you about the genes. So name of the genes, then gene description, which will tell you the, it means complete name. So which is the actinin alpha 2 for this particular gene. And now it will tell you, the next section will tell you about the evidence, that at what level information is present. So whether it is at the RNA level or protein level. So in this case, the evidence was available at the protein level. So whenever you will keep your cursor on this particular square box, then it will tell you that at what level evidence is present. So mostly these green colored check boxes represents the evidence at the protein level. Then for all the 12 sections, it will contain the information. So this actinin alpha 2, although this gene is expressed in the heart muscles, but if you click on the tissue section, then it will also tell you that in which other tissues it is present. So because it might be the tissue specific or it might not be tissue specific. So in that case, for other tissue information will also be given. Similarly, for the brain also it will tell you that whether it is expressed in brain, and if it is expressed, then in which part of the brain. So in this case, they are writing RNA, which means that for these particular tissues or for these particular sections, only RNA level evidence is available and not protein level evidence. So in brain, similarly for the single cell also RNA level evidence is available, for the tissue cell also. And for pathology, you can click on pathology to find that to which disease this particular gene is associated. Similarly for disease, so in this N is written, which means that not any, that means not any, it was not identified in any of the disease. Similarly in immune, so immune cells also RNA level evidence is available. For the blood, they have mass spectrometry data, so which means that protein level evidence might be available. Then similarly for the subcellular distribution, they have for subcellular information, they usually take the imaging data. So in that case, uh, so the pictures have been collected and in that case they identify that this particular protein is expressed in which cellular compartment. And for cell line also RNA level data is available and its structural information is also available. So you can also find the structure of the protein which is encoded by this gene. And similarly, interaction information is also given. So in this way, if you want to study any of the tissue and all the genes expressed in that tissue, then you can simply click on that particular tissue and it will give you the list of all the genes along with all the informations present at the level of 12 sections. Yeah, so this is about the enriched genes. And then in the bottom of this tissue section, you will find human subproteomes. So other than this particular proteome, you can also explore the subproteome level, which is the tissue specific proteome, the housekeeping proteome, the regulatory proteome, the secretome and membrane proteome. So in this, they have further categorized these protein coding genes into multiple sections. For example, if there are some proteins which have the housekeeping function, which has to be expressed always, which has to be turned on always. So they come under housekeeping and it will always be expressed. And then regulatory proteome. So if some protein has a role in the regulation of some pathway or any regulatory role, so it will be categorized under regulatory proteome. Similarly, some membrane proteins or secretory functional proteins, they will be categorized under this secretome. Similarly, isoform proteome. So till now we have seen that gene level information is given and its structure is given. But what happens is that there are other mechanisms which are also ongoing. So a single gene can also give rise to multiple isoforms and ultimately multiple proteoforms as well. So in that scenarios, we can go to isoform proteome. For example, if one gene information I have and I want to understand or I want to study that which other proteins it can, which of the all types of protein it can synthesize. So in that case, I can go to isoform proteome. It will tell me all the isoforms which are means expressed by that gene. So in that case, I can target them or I can study them in detail. Then the cancer proteome and the druggable proteome, for which the drug target information is available and which are the drugs which will attack those targets. So all those informations come under subproteome. So now next section was brain. So now in brain section, it will tell you the information about all the proteins which are specifically expressed in human brain. And this is the home page. So in the home page, firstly, they'll elaborate you a little about the what does the human brain proteome or protein atlas means, and from where the data is collected, and what how many regions specific data is present. So in this case, the brain was divided into 13 main regions and about which information is present. And here are the example of the latest genes which have been identified. And similarly, it will also tell you that what you will learn from this particular means human brain atlas. So what information can you get from here? So this is about it. Now, if you will scroll down further, then you will find. So firstly, they'll mention about the different brain regions as well as the different human brains. So as we discussed, then that in this brain section, it is dedicated to mammalian brain. So in mammalian brain, they have specifically looked into the human brain, mouse brain, and pig brain. So if your interest is human brain, then you can click on this human brain. Otherwise, depending on your interest and which information you want to get, so you can similarly click on any of these links. Then brain cell types. So on the basis of different brain cells, also the data has been categorized. And pituitary gland. So if you want to specifically focus on one particular part, then you can click on it. So in this, they said that they have summarized the data into 40 main regions of each mammalian brain. Now let's see, what are these 14 anatomical regions? So they are cerebral cortex, hypothalamus. So similarly, these are the 14 different regions of the brain. And if you want to specifically focus on any of the particular region, you can simply click on that particular link. So these names, if you keep your cursor on this particular information, then it will tell you some kind of genes, how many genes are expressed. And if you click on it, then it will take you to the another window, which will tell you the list of the genes which are expressed in this particular compartment or this particular region of the brain. Now, if you further scroll down, then it will give you a brief summary that how many proteins have been identified and how they are categorized. So in this case, in some places they are calling as the 13 different anatomical regions, whereas in other cases they are also writing it to be 30. So it depends that how many sections the evidence is available. So in this case, after identifying all the proteins, so in this case, 2,197 genes were classified as brain elevated genes. 97 were only detected in the brain, which means these are specific to brain. And in total, 15,331 of the human protein coding genes are detected in the human brain. So this is the same number as which was previously mentioned. So these are the old proteins, and if we divide them into multiple groups or multiple categories, then we'll have this pie chart in which 2,197 are elevated in brain, which means these genes are overexpressed in brain, and then elevated in others but expressed in brain. So these are mostly expressed in brain, but they might have the greater expression in some other particular tissue. Then low tissue specificity but expressed in brain, which means that they are not specific to brain, but they are expressed at some quantity. And then this represents not detected in brain and not detected in any tissue. Yeah, so yeah, similarly, if we go down, then it will also tell you the detected as well as regionally elevated number of genes. And if you click on these things, then you will be taken to the page where all the information will be present. So in this way, in Human Protein Atlas, all the information is given and it is mostly represented in the form of tables. And each thing which is mentioned in the light blue color, you can simply click on it and the information will be given to you. So similarly, for all those regions, the information will be given for human, pig, and mouse brain. Yeah, so now we can also search for any gene specifically. For example, if I'm studying some gene, so I can also find it in the section of brain. So not exactly on the brain only, we can find it on all the sections. So what we have to do that in the search bar, we simply have to write the name of the gene. So we might have the gene name or gene ID, anything we can write, and we'll click on the search. And then it will find that gene. So in this case, ZNF451. So for this, only single means after putting this query, only single result came up. But in your case, it is also possible that similarly means other genes could also come up which have a similar name. So in that case, you have to select the precise gene for which you want to find the information. So for that gene information will come in this particular form, and you can explore it at any of these levels. So in this case also evidence is available at the protein level. And so here RNA is written, so which means that for these tissue section or the brain section, single cell, for all these sections, they have the RNA-seq data information, not the protein-seq information. Similarly, structure and interaction information is also available. So if I want to find that whether this gene is expressed in brain or not, so I can simply click on this particular brain section and it will tell me the information about the brain. So firstly, in the journal information, it will tell me the name of the gene, human gene, and the mouse gene. And if this particular gene is also present in the pig brain, so it will also give me the gene name in the pig as well. And then firstly, the information is given about the gene name. Then it will tell about the location, that what is the predicted location of that gene, whether it is expressed intracellular or extracellular or membrane. So this is the location. Then there are IDs. So in ensemble, the gene ID is given. So in ensemble gene ID, this is the mouse gene and this is the pig gene. So in ensemble, the information is available of this gene. So that's why they have given the gene ID. And then antibodies in the assays. So whenever they were performing assays, so whether the antibodies were present or not. So in this case, no antibodies in assay. Similarly, if you further see in the right side, so it will tell you about the brain expression cluster. So in which part of the brain it is being expressed, that information will be given. In tissue specificity, it will tell you that whether it is tissue enhanced, tissue specific, or it is expressed and it is not specific to this particular tissue. So all those information will be given here. And then regional specificity, so whether it is a low region specificity or high region specificity. So in this case, the ZNF 451 is having low region specificity. It is not specific to any particular region of the brain. Then tau specificity scores are given, and then regional distribution, so detected in all the regions. So in this case, we can explore that whether the gene of our interest is expressed in any of one single region of the brain or whether it is expressed in all the regions. So in that case, we can find that information in the brain section by simply clicking on the brain section, and then this information will open up. And we can also find its structure interaction. So they are in the structure interaction sections. And if you further scroll down, then it will also tell you that in which regions of the brain it was expressed, and also the NPPM, which means the number of transcripts per million. So this is just used to quantify that at what level transcripts were identified. So we talk about ZNF 451. So this particular gene is mostly expressed in the cerebellum in the higher quantities, that is about 80 TPM, that means transcripts per million. So there are some measures to identify that whenever the experiments have been done, so how many transcripts were identified, how many reads were captured. So in that case, this is overexpressed in cerebellum. And then in white matter also, its expression is high. So they might have some criteria to identify that when to call a gene specific to particular region. So in this case, this might not satisfy the condition. That's why they have not mentioned that it is specifically expressed in cerebellum. So its expression is observed in almost all the regions. There is no such region where its expression was not observed. So that's why we cannot call it specific to a particular region, because specificity means that it is only expressed in that region. So in this case, you will be able to identify their expression as well. What is the expression profile? Similarly, if you further move down, so you will identify firstly in the human brain that if we talk about human brain, so what is the expression? So firstly, in this case, they are telling about GTEC, so which is the gene tissue exchange. So in this case, the normal proteins which are expressed, about that information is present in the GTEC dataset. So from GTEC, they are telling you about all the proteins which are available in GTEC, which means they are not considering the proteins which are expressed in the cancer tissues or which are associated with the disease. So in this case, they are only focusing on the normal proteins which are expressed in the normal tissues and which are not linked with the disease. So in that case, uh, in the GTEC also, the information is if we talk about their expression information, so it is expressed in all these regions of the brain. Similarly, from Phantom 5 human brain cage dataset, so let's explore what the GTEC means. So in GTEC, which is the genotype, sorry, not gene, but genotype tissue expression project, which examines that how the genetic variations impact the gene expression across various human tissues, and it provides a valuable resource for understanding the relationship between the genes and disease. So mostly it contains information about the normal genes, not about the disease genes, although it might help you to examine the variations because whenever you have the information of the sequence, so you can compare them with the GTEC genes so that you can identify where the variations are present. Similarly, if we talk about Phantom, so it is the functional annotation of mammalian genome 5, and it focuses on mapping and annotating the functional elements of the mammalian genome, such as the active promoters and enhancers. And this project offers insights into the gene regulation and expression across different cell types. So in this case, mostly Phantom contains the expression level information. So similarly in GTEC and Phantom, this is simply telling the expression of this particular gene in these regions. So Phantom is about dedicated to expression and GTEC is about the normal expression of the genes. So if my gene of interest is present in the GTEC, so in that scenarios, the information will be given. Otherwise, this information won't be given if it is associated to disease. And what other thing can happen is that there are some genes which have some mutations or variations, which are means with those variations they are linked to disease. So in that case, also in GTEC, the gene will be present, but without any variation. So that information is present in the GTEC, and in Phantom it will have the information of both those, means all the possible gene types or gene variant information, and their expression information is given in Phantom 5. So we won't go into the detail that what does it mean, but simply they will tell you about the expression profiles which are available in GTEC as well as in Phantom. And then they have also given the information about the pig brain. So in pig also, there is a dataset HPA, Human Proteome Atlas pig brain RNA-seq dataset. So in that case, they are telling you about the expression of this gene in different regions. So yeah, it is expressed in all the regions. So we cannot say that it is specific to any one region. And similarly for the mouse brain also, there are two datasets. One is HPA, which is Human Proteome Atlas, and another is the LN mouse brain, which is the IASH dataset. And here is the description about them. So HPA is the RNA-seq tissue-specific data, which is reported in the terms of number of or normalized expression of transcript per million, and which is present in the Human Proteome Atlas, whereas the LN mouse is the another repository which contain the expression values based on in situ hybridization available through a ABA API. So this is the another data set which is hosted by the LN Institute for Brain Science. So what is the purpose of considering GTEC, Phantom and these LN? So why they are considering it? Because although HPA is taking the help of the antibody screening or other experiments, but they are also trying to validate their analysis, and they are just cross checking that what information they are providing, whether it is similar in other or whether there are some changes, because there are some expressions which might not match. So for that reason they are providing that how the expression varies among these two data sets. So for example, in this case, you can see clearly that in HPA mouse brain, the expressions are higher, whereas in the LN mouse, they have some another criteria for measuring the expressions. So in this case, the expression energy units are given, and in this case, NTPM is given. So in this case, there is a huge variability among different regions for the expression energy of this particular gene. So yeah, this is the evidence for the mouse brain. And you can also find the what are the number of genes and what is the NTPM value exactly by just keeping your cursor on any of these x-axis labels. And similarly, if you further scroll down, then it will also tell you about the means if it is a part of a cluster. So there are some genes which are expressed as in the form of a cluster, which is expressed with all other genes, and they all genes form a cluster. They are expressed together. So in this case, if my gene is a part of any cluster, so this information will also be available, which is expression clustering and correlation. So in this case, it is expressed in the oligodendrocytes, and it has a mixed function with confidence some confidence, and you can click on I to further read about what this confidence means. And there are 601 genes in this cluster. So which means that all these genes function together, they are expressed together. So that's why they are categorized into a cluster 28. And this is the means so after dividing it into different clusters, so this particular cluster is highlighted, which contains my ZNF451 genes. And you can go to the interactive expression cluster page as well, which you will see further. So 15 nearest neighbors based on RNA, brain RNA expression. So in this case, these all are the neighbors of my gene, which is ZNF451. So in the cluster, there are some other genes which are also available. So how they identify these clusters? There will be a network which they will form. So that's why they are calling them as neighbors. That means these neighbors are also expressed with this gene. So they all form a single cluster. So in this case, you can also find that information as well, that which are the other proteins which are expressed with my gene of interest and what is the correlation. So in correlation, it will tell you that if my gene is overexpressed, then whether that is also overexpressed. So is there any link between the expression of both of them? And then here is the name of the clusters. So in the human genome, all the genes which are present, they are divided into multiple clusters. So similarly, this will tell me about the number of means cluster serial number. That what is the serial number of my cluster? It does not mean that a number of gene in the cluster, but serial number for that particular cluster. So it is 28. So for the entire brain, they have made different gene clusters, and that way it was 28th cluster. And if we click on the interactive expression cluster page, so it will open up in this way. So as in the week 3 or 4, we were discussing about the dimensionality reduction. So similarly, UMAP is also the another way of dimensionality reduction. And this is simply used to divide or categorize the data into different categories. So in this case, blood and immune cells, endocrine cells, endothelial cells, glial cells. So this is not specific to that ZNM451, but it is the interactive page that how different gene clusters are divided and in which regions or which tissues they are expressed. So in this case, they mentioned that there are 56 gene clusters which they have identified for entire human brain, and these colors represents the particular cells where those gene clusters are expressed. So UMAP stands for Uniform Manifold Approximation and Projection, and it is a dimensionality reduction technique, which is simply used to represent the high dimensional data in the low dimensions while preserving its structure. So in this case, they will identify that they will only capture those genes which are varying among different cell types or different regions. So in that way, the specific genes have been mentioned. Now in the bottom, if you further scroll down, you'll find these are the IDs. So these will be the cluster IDs, and then main specificity, which will tell you that which region of the brain or which particular tissue it is majorly or specifically it is expressed. And then about the function, so mixed function and number of genes which are present, and then their annotation reliability. So means there are some cases where annotation might not be accurate or there are some confusions with the annotation. So in that case, it will be low. And if it is medium, which means that you can rely on this information, and it might also be high, which means that you can confidently accept this annotation. So similarly, you can also identify those genes which are expressed in the cluster in particular region of the brain or particular cells. So in this case, the cell level specificity information is given. Yeah, so this is all about the ZNF451 information, and you can also go to the summary in which all the summary will be given. So instead of going to all these sections one by one, you can simply go to summary. You can simply click on summary and it will just give you the overview, entire overviews. So it will tell you about the protein, gene name, protein class, to which class it belongs, and what is the evidence. So whether evidence is available at RNA level, protein level, or gene level. So that will also be given. It will also tell you number of transcripts. So as we discussed that one gene can also give rise to multiple transcripts. So in that case, that information you can also be explored. Protein interactions, you can also identify its interacting proteins. And tissue profile not available. Subcellular location, it is localized to the nucleoplasm. So if you want to identify subcellular location, you can simply go to summary and you will find all these informations. And then it is also telling you about the tissue specificity, tissue expression cluster, brain specificity. So means to get all the information in one page, you can simply go to summary. And if you want to get into more details, then you can explore these sections one by one. So this is about how you can identify one particular gene in this Human Protein Atlas and what information you can explore from Human Protein Atlas. And similarly, if you scroll down, then there are other sections also, such as immune cell, cancer cell lines, whether this gene is expressed in cancer or not. So all these informations will be present. Then similarly for the blood as well. And what are the functions? So annotations are also available that given gene, what function it performs, and what is the molecular function, what is in which biological process it is involved, what are the ligands which bind to this particular protein, and this is the summary. So you can also identify the function, so which is already for which the information is already available. And similarly, if you go to section overviews, and then for each section there will be a small overview which will be given. So not entire detail, but specific details will be given for each section. If you go to section overview on the left side after completing your search, yeah, similarly for all the 12 sections, this information will be given. Then you can also go to gene information. So for example, your focus is that you want to identify the information at the level of gene. You want to just look that in which chromosome it is present, what is its cytoband, and what is the exact location of this particular gene in the chromosome. For example, in this particular week, you have also looked into the integrative genome viewer. So in that case, you can simply get the information of the coordinates in the chromosome, that where this gene is present. So you can capture it from this particular section, and you can simply go to integrative genome viewer and you can put this information of the chromosome and this particular coordinate information, and you will be able to look that gene in the genome where that sequence is present and what are the three-frame translation of that particular genomic region. And you can also compare it with the variants, that what are the variants which are present. So in that way, this information can be obtained from gene information section. It will also tell you the ensemble ID. So these are also the links to other repositories. So ensemble is also a big database which contains the entire information about the human genome, different genes, transcripts. So in this way, and then Entrace gene. So in this, Entrace is hosted by NCBI. So it will take you to the NCBI, which is another repository, another database. Then HGNC, UniProt. So these are all other databases for which the information is present here. So these are also kind of providing you the links that if you further want to explore these repositories, so you can go to them and see that what information for your gene is available in these. Then protein functional information is also available in the gene information section. Yeah, and similarly, if you further move down, then protein information will also be given in brain information only, which will tell you about all the protein transcripts. So as they mentioned, so there are different transcripts for this single gene. For all these transcripts, information is given, whether it is obtained from SwissProt. So these are also the protein level databases. SwissProt comes, SwissProt is the dataset which contains the protein sequence information or protein information which is coming from experimental evidence, whereas Tremble stands for translated EMBL, which means that it is translating your RNA information to protein. So this is not experimental information, but it is customized, that what are the expected proteins which can be synthesized from the RNA. Then protein class, gene ontology, length and its mass, signal peptide, whether it is a signaling protein, signal peptide is present or not, whether there is transmembrane region. So transmembrane region will tell you that whether my gene of this any of these proteins are expressed in the transmembrane region. So similarly, all this information can be studied from this gene information compartment or gene information section. Then if we go to RNA data, so it will tell about the RNA level data, that how means after capturing the RNA level data, what are the distribution of, what is the expression of the RNAs in different tissues and all these tissues. Similarly, if you further scroll down, then GTEC tissue dataset, TCGA, which is the for the cancer genome atlas. So and then GTEC is for the normal genotype tissue expression. So normal in the normal, how is the expression is present, and in TCGA in the cancer cells or in the cancer tissue specific expression of this particular gene. So as we can see, it is very low, which means that this gene is not specific to any of the cancer. And now the last one is the antibody data. So if you want to know that whether any antibody is present to my gene of interest or not, so you can simply go to antibody data and it will give the information of the antibody. So which antibody they have used to identify this particular protein and which can further be used. So in this way, you can find all these informations from here. Yeah, and you can also download the information as well. You can download the data. It will, you can either download it by using some programmatic access. You can simply write it on the terminal, or you can simply download also from these links which are provided. You can also download them in the archive form. Then for the normal tissue, how in the normal tissues this gene is expressed, because there are some genes which are also expressed in cancer tissue. So it depends on you that which information or which expression you want to capture. So in this way, all the links will be given in the bottom. If you further scroll down, then the downloadable data information will also be given. So you can download it as well. And then now let's talk about the another section, which is the subcellular section. So till now we have explored the tissue section and the brain section, although we have looked into other sections as well by taking the ZNF example. But other than that, we can also go to subcellular section. And why I'm discussing this, because in this only we'll find the organelle level expression information, because there was some question in the assignment in which I think you have to tell about some organelle level expression, means which was related to this. So for this, we can simply go to subcellular section, and firstly in the home page, the information will be given that how many genes have been identified and what you can learn from this subcellular section. Then if you further scroll down, so it will tell you about the cellular and organelle proteomes. So there are some proteins which are multi-localizing, which means they can be present in multiple organelles, which will be present throughout the tissue, throughout the cell. Or there will be some proteins which are organelle proteome, which means which are specific to some organelles. And then cell cycle dependent transcriptome and proteome. So which are cell cycle dependent, which are expressed in some particular phase of the cell cycle and which might regulate the cell cycle. So those informations are also given in this. So if you further scroll down, then it has divided the information into the different sections: the nucleus, cytoplasm, endomembrane system, secretory. So in this case, the proteins are divided into these four categories. So firstly, the proteins which are expressed in the nuclear membrane, nucleoli, nucleoplasm. So that information you can find here. Similarly, for cytoplasm, different uh, compartments in the cytoplasm for where the protein expression information will be available is the actin filament, centrosome, cytosol, mitochondria, microtubule, intermediate filaments. And when we talk about endomembrane systems, so in that case, endoplasmic reticulum, Golgi apparatus, plasma membrane, vesicles, these are the categories. And in secretory, secreted protein information will be available. And if you're interested in any of these sections or any of these compartments, you can simply click on these light blue colored names which are written, and they will take you to the all the proteins which are expressed in nuclear membrane. Yeah, and if we go to the organelle proteome, so by further scrolling down, you will find the organelle proteome, or if you do not find, then you can simply click here, the organelle proteome, and it will take you to this particular page. So in organelle proteome, you will find the subcellular localization of proteins, the protein distribution in human cells, and validation of antibodies and location data and relevant links and publications. So we will explore all these things. What does they mean? So in subcellular location, it will tell you about the where the protein means in all the entire cell in which particular location my protein is expressed and for which they are using the imaging data. So in this case, you can click on any of these organelles and you will be taken to all those proteins which are expressed in any of these particular subcellular locations. If you further scroll down, then it will tell you about the protein distribution in human cells. So in this case, the information is given about the genes which are or the proteins which are expressed in all of um, these organelles. So there are total 35 organelles and subcellular structures for which they have identified 13,147 genes with the protein localization data in the subcellular section. So in this case, you will be able to find in the cytosol there are uh, 3.8 log fold genes which are present. And similarly for all these organelles, you will be able to find the number of genes. So this refers to the cytoplasm. So as we have seen that there are different categories. So cytoplasm, these all organelles will come under cytoplasm, then these organelles will come under endomembrane system, and these uh, regions will come under nucleus. So for example, if you want to explore the nuclear membrane gene, so you can simply keep your cursor over this bar and it will tell you number of genes which are expressed in the nuclear membrane. Similarly, for other regions of the organelles also, you can simply keep your cursor and you will be able to find out how many number of genes are present if you keep your cursor. And if you click, then the another window will be opened, and which will also tell you about the all those genes, and the table will appear, which will also give you the link to all the 12 sections so that you can go into the detail of those genes as well. Yeah, then if you further scroll down, then it will also tell you the validation of antibodies and location data. So in this case, for all the other means for all these organelles, you will also be able to find the number of proteins and what is the percentage and location reliability. So whether you can rely on this information or not. So whether it is enhanced, supported, approved, uncertain. So in that way, they have divided all the identified proteins into these four categories, which is enhanced, which is 1328, which means they are enhanced in specific organelle. Then supported, which means there is a supported evidence which is available. Then orange one represents the approved. So 6689 are the approved genes or approved proteins for which you are very sure that yeah, these are expressed in these organelle. And then for 751, the information is uncertain. So sometimes what can happen is that whenever you are looking into the antibody specific data, so there are some non-specific interactions which can happen and you can identify some protein or gene by mistake. So in such cases, which is so we are uncertain that whether this gene is or this protein is specific to this particular organelle or not. So there are 751 for which we are uncertain. Yeah, so now let's move to the Human Plasma Proteome Project. So, till now, is everything clear or is there any doubt? Okay, thank you. Yeah, so means we have covered the Human Protein Atlas and different sections and how you can explore the information and what are the different sections for which the information can be explored. Now we will talk about the Human Plasma Proteome Project. So it was initiated in 2002. So it was initiated because there were some challenges which were present in the Human Protein Atlas and tissue specific expressions. So there was difficult to identify the sensitivity and analytical depth. So to resolve that, the main focus was on the plasma proteins and blood plasma specifically. So this was just initiated so that we can deal with these challenges, and we will be specific, and the data will also be sensitive because it will be obtained from the plasma. So that's why it will be systematically collected and stored and accessible through biobanks. And its ultimate aim was to collect and facilitate the advances in plasma protein analysis using different technologies, diseases, and concepts. So what happened is that to identify the proteins or genes in different tissues and then being calling that data to be reliable is a bit difficult. And for all the proteins, antibody information is also not available. So in that case, plasma proteins, it is mentioned that in plasma maximum proteins expression is higher because in blood it is easy to capture the protein. So that's why plasma protein analysis will be better to do, means it will be more sensitive as compared to the tissue specific information. That's why it came up, and it is hosted by Peptide Atlas. So we'll see that where you can find the information of the proteins which are expressed in the plasma, and then comparison of the Peptide Atlas data with CHPP. So you can means go through these links if you are interested. These are the useful links. Now we'll see that where this Human Plasma Proteome Project is hosted. So it is hosted in Peptide Atlas. So if you click here, HPP Data Central, you will be taken to this particular homepage, which will be present in the Peptide Atlas. So you can simply go to Peptide Atlas, then also you'll be able to find Human Plasma Proteome Project Data Central at Peptide Atlas. So I hope it will be clear that in this case, the evidence will be at the level of proteins because they will be collecting the peptide sequences which are present, which are expressed in the plasma. So that's why it is present in Peptide Atlas. And so in the initial lines, they complete history will be present. What is the overview of the project, when it started, how many laboratories came up and contributed, how many data sets were constructed, and how many data sets are specific to this plasma proteome project. Similarly, latest publications are also available. So whenever these data sets have been constructed, so these people, they also write a paper that how the data collection has been done. So in that case, you can also explore their publications to see that how they were able to identify and what are the steps, what are the workflows which they have followed. And then there are some other informations which are also complementing this publications. So summary table of proteins identified in the build. So these build, what does build means? That each year new data means new proteins have been identified, new things keep on adding. So what they do is that they do not mix all those informations. They keep them as a separate builds, or we can say separate versions. So that means if somebody is performing research on some data, and now I'm starting my research on the 2024 data set, for example, and what if after 6 months they add new information to that? So in that case, my data set will get disturbed because new genes will be available in that, or new proteins will be added. So in that case, what they do is they separate those builds. So that means if somebody is working with one particular data set and some results have been produced, so for anyone else to reproduce those results, they can simply go to same data set, so that similar results can be verified, that whether they are able to reproduce or not. So in that case, the builds means what is the version. So the currently in 2021, this publication has been done. So in that case, they might have means published a new build. So in that case, you can simply go to the summary table where the entire summary will be given, that how many proteins, how many genes information has been identified, how many peptide information has been identified. So in that case, and then there is a table of all the data sets that have gone into the build. So in that case, all the data sets, because there will be multiple data sets. For example, multiple tissues I'm performing experiments on. So in that case, there will be multiple data sets, and data sets which will be tissue specific, or they might be experiment specific. So in that case, it will also tell you about the data sets which are incorporated to form a build. Then direct access to browse the Plasma Protein Atlas 2021 build. So if you directly want to go to 2021 build, then you can click on this link. So this is the 2021 build, but currently there will be, I think, 2023 or 2024 build, which will be currently available. So this publication is old and they have not recently published anything. That's why they are providing link to 2021 build. So keep these things in mind, that which build you are clicking on and which build which is being shown to you. Then Human Plasma Peptide Atlas. So you can simply go to this, and this will also tell you about the Human Plasma Protein Atlas, Peptide Atlas. It will also tell you the peptides which are present. So we'll see them one by one. So firstly, if you scroll down, then you'll find the Peptide Atlas and the Plasma Proteome Project. So it will tell you about the builds and what are the different entries which are present. So in this case, I have specifically selected the 2021-7 build. So this is, in this case, the build belongs to 7-2021, which means which was available of the which is available for the year 2021. And in this case, these are the chromosomes for which the information has been identified. There are nextprot entries. So nextprot is also the another, we can call it as a repository where the information is stored. Then canonical, uncertain, redundant, and not observed. So which means the peptides which are canonical, means canonical are the peptides which are normally available, or which for which the evidence is available. So here canonical means proteins seen with the two distinct or uniquely mapping peptides. So we can also call them that for which greater evidence is available. Then uncertain means proteins with some evidence that is not sufficient for the canonical status. And then redundant means proteins that have peptides that map to them but not uniquely, thus not needed to explain the observed peptides. So what can happen is that when we say peptides, they are the smaller sequences, not as big as proteins. So what can happen is two proteins can possess a single same peptide. So in that case, it will be a redundant peptide. So you are not sure that from which particular protein it is coming. So it will be for that particular peptide, we will be linking it to both these proteins. So that will be labeled as redundant peptide. Then not observed, which means that no detection at all in the peptide atlas above, or our criteria is very stringent. So yeah, as we have seen that in mass spec, what can happen is that the peptide spectrum is obtained, but you are not able to identify the sequence. So in that case, it will be written as not observed. So in this case, they are telling you about the distribution, that in each chromosome, how many next prot entries are available and how these next prot entries can be further categorized, whether they are canonical peptides or they are uncertain or redundant, not observed. So in this case, this is specific to the blood plasma. So in blood plasma at the level of chromosomes, along with this, for the mitochondrial genome also, they have identified next prot entries. Now I'll little bit talk about the NextProt. So it is developed by SIB, which is the Swiss Institute of Bioinformatics, and it is a comprehensive human-centric discovery platform which offers its users a seamless integration of and navigation through protein-related data. So this is another repository. And here we are talking about the peptide atlas. So in this case, it is taking the data from the NextProt, which is the another repository. And this NextProt is also taking the data from multiple other sources. So means all these things are somehow linked to one another and means not as means it's not that peptide atlas is only identifying everything, but it is also is dependent on the other repositories for their data set. And in this way, there are different data sources for this. And in the next plot, you can simply search for a particular protein for which the peptides in the experiments peptides have been identified, or you can simply give your sequence. You can also write the peptide sequence, that whether in any of the experiment, whether that sequence was available or not. So you can simply search here. And there are different categories, gold or other categories will also be present, but gold means that which are highly specific, which are canonical kind of entries, for which the confidence level is high. So that is gold and which will be selected by default. But if you little bit want to relax, then you can simply select the other categories which are present here. Yeah, so if you further scroll down, then in NextProt only, it provides the protein existence by chromosome. So means what this figure represents. It these colors represents the protein evidence, that at what level protein evidence is available. So this light green represents the evidence at the protein level, and then PE2 represents the evidence at the transcript level, PE3 represents the inferred from the homology, PE4 is predicted, and PE5 is uncertain. So similarly, for each chromosome, the evidence level information will be given, that how many means the peptides which have been identified, how they are linked to the proteins, whether there was evidence at the level of protein, because we know that whenever in mass spectrometry or any experiment, whenever peptide spectrum is captured and whenever we have to map that to the human proteome, so we use the databases which are already containing the protein sequences. But what can happen is if my peptide is novel, which means it is not available in already available protein sequence databases. So in that case, we design the customized protein sequence databases from the transcriptomic data. So in such scenarios, the evidence will be available at the transcript level, which means that particular protein sequence information is not available in the databases. So that's why it is the transcript level evidence. And similarly, in one of the week you have also looked into that we can also infer it from the homology, which means that the proteins which are from the same family, so that level evidence we can also take. We can see that if given peptide is having a sequence which is relating to the means which is also available in the protein of the similar family, means it's a little bit varying, but it is showing the homology. There is some kind of similarity between those sequences, so that is known as inferred from homology, which is not the completely customized protein sequence, but it is somehow related, so that is known as inferred from homology. Then PE4 is predicted and PE5 is completely uncertain. So mostly the evidence is available at the protein level in the next prot. And if you click on any of these numbers, then it will also give you the list of all those genes which are present. So in this case, if you are studying any chromosomal level genes, so you can simply go to next prot and click on any of these chromosomes. Yeah, then if we click on the direct access to browse the plasma peptide atlas 2021 build, then we can click on this and we'll be able to go to the peptide atlas. Now in this case, it will direct me to 2021 build, but in this case, I have selected the 2023 build, which was the latest build. So each year, new build keeps on updating. So this was added in the April. And in this, if I want to find any protein or complete for the entire human plasma also, I can search for the peptides and their related gene or proteins. So if you see here, firstly, this is the search option or search menu, in which it will be giving you a form in which you can put on the entries given, means you can ask any query on basis on the desired query or what information you want to find. So first thing is the Atlas build. You have to select the build. So if you click on this particular means scroll down option, so it will tell you all the builds which are available, and from means even all the primitive builds will also be listed here. For example, 2019, 20, 21, 22. So I have selected 2023 build, which is the latest one. The Nextprot mapping. So in Nextprot, the protein sequence information will be available from where the mapping has been done for the peptide spectrum matches. So that is also 2023 512. Then protein name constraints. So if you want to find a specific protein, then you can simply write the protein name and upload file of proteins. So you can also upload your protein file. So in that case, if you have a sequence information or if you have the list of proteins, so you can upload those file also. For example, if I don't want to search a single protein, but I have a list of proteins and I want to find that whether these are means observed in human plasma or not. So in that case, I can upload my file. Then gene name constraint. So if I have information of a gene for which I want to find, so I can simply write the gene name. Similarly, description constraint. For example, if I want to find all the genes which are related to signaling, so I can simply write that description, signaling or metabolism. So in that case, that thing can be written just to specify that what is your query to further filter out or get the means filtered out results. So for that case, you can specify it with each and every section so that you do not get all the protein information which is available. This is just to shorten the results which you get and precise results. Similarly, you can also select the chromosome. For example, if we are interested in chromosome number 4, for example, so I can simply write chromosome 4. So in that case, all the genes which are available in chromosome 4, their information will be given. Then start position in chromosome, so that also we can verify, end position, that also we can specify. Protein evidence level, for example, we are only interested at protein level evidence, so that we can also select. Peptide atlas category, so that also we can select that whether that peptide should be canonical or it should be identical or it should be indistinguishable. So in that case, these all comes under the query section, and this help us to make our search better. So after selecting all these options according to our desire, we can simply click on query. And if you do not put any of these options, then what result it will give you? By default, it has selected PE, protein level evidence, and canonical. So if you query it, then it will tell you about all the proteins which are expressed in human plasma, because we are not giving any specific information. So if we click on query, then this particular window will open up, which will give you the list of all the genes and their next protect sessions, and then ensemble gene IDs, refsec gene IDs. So these are the other different repositories where the information is present. So it is just telling you their IDs. So because there are some genes for which the information is available at ensemble as well as at the refsec. So in the refsec, they are telling you about NP, which is the ID given to the protein sequence. So in this way, they are providing you the multiple IDs, so that if you get the sequence or protein sequence, then you can also verify it with the refsec as well, and ensemble also that how that sequence is available in this. Then chromosome, that which chromosome that particular gene is present, what is the start coordinate, end coordinate, and N chromosomal locations, next protein evidence, peptide atlas, means what is the category of that particular peptide, and then number of distinct peptides which are observed for this gene. Because one gene, it is not that only one peptide will be identified. There are chances that multiple unique peptides are captured in the mass spec. So in that case, it is also telling you about number of peptides. Because more the number of peptides, you are more sure that this is the gene and this gene is overexpressed. That is also another thing. And then in description, it will tell you the description of this particular gene. So that in this way, you can explore the human plasma proteome project dataset. And then in the summary, so firstly, in the form, it is about the query where you ask a query. In result set, it will be about the result, and if you click on summary, then it will be a brief summary. So it will tell you about the canonical proteins for which the peptides were canonical, more than two peptides were present. So in that count is the number of genes. So that is 4,603. And percentage will tell you that out of total genes, how many genes were canonical. So in this case, it is 100% total and percentage. So in this case, all the genes or peptides, sorry. So in this case, genes and proteins are used means here and there. So in this case, protein identification, so we mean it as like 4603 is the number of genes also and proteins also. It means the same here because they are not talking about the transcripts or isoforms. They are simply talking about the genes for which the proteins have been identified. So these 4603 represents the genes where these peptides were labeled as canonical peptides. So this is about the summary. And then if we click on Human Plasma Peptide Atlas, then it will take us to this homepage, which will be having a short, brief summary. So in this case, firstly, they will explain that what this Human Plasma Peptide Atlas provides you. Then they will tell a brief summary of the build, which is the latest build. So in this case, the latest build was 2023-04, which means in this tells about the month and this tells about the year. So means when this data was previously means last updated. In this case, there are 113 data sets from where the different informations have been merged. 330 new experiments have been done. Then MS runs, so 35,801 MS runs were done. Then peptide spectral matches. So these many peptide spectral matches were identified, and about 2,63,150 distinct peptides are present in this dataset, and 4,608 canonical proteins are the canonical proteins which have the major peptide level confidence for those proteins. Yeah, and in the means bottom of this particular homepage, you will find the search option. So in this, you can write the identifier or you can write anything in the text. For example, uh, if you have the name, for example, hexokinase. So in that case, you can simply write the hexokinase instead of mentioning its identifier. And then in peptide, you can also means query your peptide sequence and you will be able to find the protein to which that peptide sequence belongs to if that is available in the experimental data set. And then you can also enter the sequence, which can be a protein sequence or peptide sequence, and you can query it so that it will identify it against these this particular build. And in all these data sets, that sequence will be searched and its respective information will be given to you. And if you further scroll down, then you'll be able to browse the information. So, for example, if you do not want to see all the peptides, then you can be specific for the canonical peptides, which have the canonical level evidence. So, in that case, also you can click on any of these options. Yeah, and then another thing which I want to mention is that they have mentioned that in Human Plasma Peptide Atlas, it is available human plasma datasets were reprocessed from the raw files using the transproteomic pipeline. So from the raw files, how they have identified, so for that they have used the transproteomic pipeline. And if you click on this transproteomic pipeline, that how this pipeline works, so it will be it will open up like this. Firstly, they have explained, and then how this pipeline works. So what it takes is it takes the raw mass spectrometry data, which will be taken by MS Convert, which will be MZML data. And then peptide identification is done. So for peptide identification, there are different tools which they have used. So such as Comment, Extendum, SpectraST, Kojak, Sequest, Mascot, all these tools have been used for the protein identification. And then peptide validation is done by the Peptide Prophet, iProfit, and PTM Prophet. Then quantification is done by these tools. And then for the protein assignment, they have used Protein Prophet. And ultimately, protein list is obtained from the C-beams, S-beams, and Proteographer. So these are different tools which are used in the Human Plasma Peptide Atlas for mapping the peptide spectra to a protein sequence or the protein level information. So this means that they have used multiple tools so that their results are more accurate. So this means this is what meant by the transproteomic pipeline. And you can also read their paper if you are interested that how they have done this analysis. And then there's a little bit of literature about the HUPO's Human Plasma Proteome Project because it was started by the HUPO. And then there is another paper which is the plasma proteome profiling to detect and avoid the sample-related biases in biomarker studies. So these two papers they are quite means helpful and educational. If you want to read, then you can go through them. And from this particular report, I have found a table in which they have mentioned that how you can reduce the biases. So journal instructions are that avoid pooling of samples and use plasma or serum exclusively and not a combination. Then how the sample collection should be done. So in this they have mentioned that how the experiment should be designed so that you can increase the accuracy and you will be able to identify the biomarkers. So that means in the end the evidence or the means we can call them as the validated and we should not be uncertain about the identification of any of those proteins. So this is about the how what things we should keep in mind while performing these experiments so that bias can be reduced. So you can read both these papers if you are interested. And now we'll move to Integrative Genome Viewer. But before moving further, I'll just check that if you have any query. So till now, is everything clear? Okay, thank you. Yeah, so now we'll talk about the Integrative Genome Viewer. So as the name suggests, that firstly it is an integrative kind of a thing, and in this you can view the genome. So in this you can view the genome sequence, and you can also decide that at what level or means you can adjust the scale. You can also see all those genes in this, I mean, a smaller way, or you can even scale it and go to the level of single gene. So that is about the IGV. And if you do not want to install the desktop application, then also it is fine. You can use the web app. And in the web app, you do not have to install it. You can simply click on it and you will be taken to the IGV homepage, which will be online server. And you can simply do all those things which are done with the offline app. And then there is JuiceWorks Web, which is the high C contact map viewer. So if you're interested, then you can go through this. But otherwise, IGB web app is sufficient. And then for developers, they have given some of the files. And for citing, for example, if somebody is performing a research by using the Integrative Genome Viewer, then they can cite it by these publications. And if you want to read more about IGV, then you can also go through these publications. So what is Integrative Genomic Viewer? It is a high performance visualization tool. So it is a visualization tool where you can visualize things for the interactive exploration of large genomic data sets, including the sequencing data. So this will also provide you advantage. For example, if you have the two different sequences, then you can also compare them and you can also find that where are the variants available, at which position it is available. So all those things can be simply done with the IGV and it enables the researchers to view and analyze genomic features in a detailed and flexible way. So in the later slides, we will see that how flexibly we can do this. So firstly, if you open that IGV by clicking on the web app, then the home page will appear like this, in which firstly it will mention about all the menus will be about genome. So in this case, you can select the genome builds, then tracks, sample info, sessions. You can share it. You can also put it in the bookmark. You can also save the image. For example, if I'm exploring chromosome number 5 and all the genes which are present in chromosome number 5 are shown here, so I can simply save that image also instead of taking a screenshot. Then I can also view these information in a circular view. So instead of looking at the linear chromosomal distribution, I can also convert it into a circle for each single chromosome, or depending on us, we will be able to see it in a circular view that which of the genes are present in which chromosome and at which position it is present. And for the help, for example, if you want to get any help or you want to check that what we can do it with it, then you can simply go to help. It will tell you. So in this case, by default, it has given the G19. It depends that. So you also have to check that which genome build is given. So as in previous case, we have seen in the plasma protein project also, we have seen that each time new build is present. So similarly in genome also, uh, means each time whenever the new genome assembly comes up, whenever new experiments have been done, so new things of the genes have been explored. So in that case, each time new genome assembly comes. So HG19 is nothing but it is telling the about the genome assembly, that after performing the sequencing of the entire human genome, at in which year, means year wise different, not just year wise, but whenever the new there are changes in the entire genome sequence, so new genome assembly is added in which new genes have been identified. So in that case there will be different genome assembly numbers. So in this case it is HG19, and we'll also see that what other numbers are present. Then in this next, this particular section tells about the chromosome, that if you want to focus on any of the chromosome, then you can select from here. And along with these 22 autosomes, there are X and Y chromosomes also, for which you can identify the genes, and you can also see the sequence. Then in case of all, it will tell you about the all the region of that chromosome. So in this case, you can enter the coordinate or you can enter the positions, that in that chromosome, from what should be your start position and what should be your end position. If my interest is about some gene, then what I can do is I can simply write its coordinate, that in this particular chromosome this is the location of my gene, and I simply want to see that sequence of that gene, so I can put my coordinates here. So how we can further explore the data here? So first step is that we have to select the genome assembly version. For that we can click on the genome option. It will open up this pop-up bar, and in this we can select, so for example, local file, Dropbox. So if we already have a genome sequence, we have already sequenced the genome, then simply we can select any of these files. Means we can select our local file from these options, or if my file is available on Google Drive or from UCSC genome browser also I can select. Otherwise the other options are human HG19 build, and then HG38 is the latest one which is present, and means they have given HG19 because this is by default build which is shown here, that's why it is given here. Otherwise I can also go for HG38. And then now currently what happened is that previously, whenever the human genome was sequenced, then it was not sequenced till the telomeres. Only the coding region was mainly sequenced. But now the newer sequencing techniques have also helped us to identify all the known coding regions as well. So that comes under the T2T, which means telomere to telomere sequencing. So in this case, it will also encompass the genomic sequences, which will correspond to known coding regions as well. So now these are the latest, means if you want to get more precise information, then you can go to human T2T, which contains telomere level information as well. So yeah, you can go to GRCH38, HG38. So in this case, I've selected this. And in your assignment, there was also, I think, one of the questions. So in that case, they have asked you about the HG19 build. So make this thing very sure that whenever you are performing the analysis or visualization, and make sure that what is the current build, which is means for which you are seeing it, because in each build or in each assembly, the information might change. It might vary. So in this case, I have selected this build, which is the latest one. The next thing you have to do is you have to select the chromosome number, because it will help us to visualize it in a better way, because it's very difficult from this how you will analyze it. So we can just restrict to one of the chromosomes. So in this case, I've selected chromosome number 4, and then you have to identify or you have to put some chromosomal position coordinates, so that you can restrict your search to that particular sequence. So in this case, I have selected these coordinates. I think this was also taken from your assignment only, but my results will vary because I am focusing on HG38 build, and you have to do it with the HG19 build. So keep that in mind. Now in the next thing, they are showing you 61 base pairs. So which means that this will encompass 61 base pairs. 61 nucleotides will be present in the sequence. And after selecting all of this, we will click on this magnifier and it will show that particular sequence. So firstly, it will tell about the chromosome, that this is the chromosome number 4, and in this chromosome number 4, I am specifically focusing on these coordinates. So 879 will be from here, from this G, and then all the nucleotide sequences are mentioned till the point of uh, oh sorry, it is 7 crore 3 lakh 18,939. So it will be means here. So that is being hidden because I've kept it. Yeah, so now this will only tell you about the DNA sequence. And now you also want to look into the protein sequence because you're interested at the level of proteins, that what are the amino acids which are being encoded from this. So what you can do is you can click on the this setting option and it will tell you about the reverse and three frame translate. So what it will do is for this particular genomic sequence, it will translate it into three different frame. It will translate in three different frames. So for example, when we start from this position, then from the next nucleotide position, and then from the next nucleotide position, so there are possibly three different frames. It means there are only three frames in which we can construct or we can synthesize a new protein sequence. So I'll just explain it. Yeah. For example, if this is my sequence, then how I can, because we know that triple nucleotide bases form a single amino acid. So how many different ways I can change my protein sequence? I can simply start my protein sequence from G, then it will be GAA codon, first codon will be GAA. Second option is that I start my sequence from A, so it will be AAA, that will be second codon. Means that will be my second frame, first codon, and then third is AAA, which will start from third position. That will be my third position or third frame, first codon. Now, when I will jump to my fourth frame, so it will be AAT. So this particular sequence will also be captured when I start from GAA. So in this way, only three unique frames or three unique sequences will be can be captured. So that's why there is only three frame translation. And when we say six frame translation, what we mean by that? That from both the directions, from front direction also and from the reverse direction also. That's why we call six frame translations when we, as we have seen in BLAST also, that translated BLAST. So that comes under that three frame translations. In this case, these are the translations from the DNA sequence, the translations which are, means you can also call them as the customized, that what are the possibilities. So there are three different protein sequences which you can see by clicking on the three frame translate. And if you want to reverse it, then it will also be reversed. And then also for that reversed sequence, that positive strand or negative strand. So on both the directions, you can see that what are the possible protein sequences which can be synthesized. So in this case, we are only focusing on the three frame translation. Now, what information you can get from this particular section? So these red colored means rectangular boxes tells about the stop codon, where the stop codon is present, and these green boxes tells you where the start codon is present. So as we know that ATG is a start codon, ATG in the terms of DNA and AUG in terms of RNA. Because it is my sense strand, which so sense strand is the one which is opposite to the template strand. As you, I think you will be aware about the transcription. So whenever my DNA is double stranded and there are two strands, one is template strand and one is sense strand. So sense strand is the one which is complementary to template strand, and template strand is the one which is for which the RNA is complementarily synthesized to that strand. So which means that my sense strand of DNA will be exact copy of my RNA. But the difference will only be that T will be replaced by U in case of RNA. So now in this case, they are telling about the sense strand, which means that by looking at my sense strand, I can simply predict my protein sequence because this will be exact copy of my RNA. So that's why they have taken it. So ATG is the start codon, whereas TG is the stop codon. So in this case, they have labeled it that where the possible stop codons could be present and where the possible start codons could be present. So similarly, the green boxes are telling you that. Now, what are the observations that can be made? First is the genomic coordinates. So in this case, these are the genomic coordinates which you can see. Then there is a sequence data. So this is the DNA sequence level information. GAA, these are the nucleotides. And then there is three frame translation. So these are the three frame translations. And as I've discussed, the red blocks means the stop codon and the green blocks means the start codon. So here, if you see, there are multiple stop codons, which means that this particular region is not actively coding for any of the genes, because as you can see, there is no start codon in between. Only stop codons are present, which means there is no space for protein coding ability. So this region is not corresponding to any of the gene. is this particular sequence in the first frame. Um, yeah, and then about the track information, the refseq genes track indicates the position of genes or the coding sequence within this genomic region. So yeah, and you can also increase this. So this is just to, for example, if you click on minus minus, then it will kind of shrink. Means these boxes size will reduce. So this is just to scale up the visual or the figure which are shown here. Yeah, so now what I have done is this red line, if you can see here, is the region which you are currently viewing. So what you can do is that you can also move this with the help of a cursor. You can also move this particular red bar here and there. And if you move it, then you will be kind of moving in the entire chromosomal sequence. This is the entire chromosome, fourth chromosome. And you can, if you are moving it, which means that you are moving to different regions. So in this case, I have moved down and I have found one gene which is MAN-BA. So this is the MAN-BA gene which is already labeled in the IGV. In this case, I'm not completely showing this IGV, but this is the location, exact location. It starts from 770 to 452 and it is present on, it is the negative strand. On the negative strand, this is the location. And in this case, I'm focusing on this particular coordinate position. So what you can do, if you move this red bar, then you will be able to find the regions where the genes are present. And for each gene, they will also tell you about the exact location and the ID of that gene. So in this case, I'm focusing on MAN-BA. So in this case, you can count the number of stop codons and the start codons which are present. So out of them, one of the sequence will be the ideal protein sequence for this. These are just the possible three-frame translations. So as 452, yeah, so which is not shown here. But if you move further down, then you'll be able to find it. Yeah, so in this way, you can also explore for each gene that from which position to which position it is present and in which direction that particular gene sequence means for that gene, in which direction the protein sequence is obtained, whether it is in the positive direction or in the negative direction. So in the forward or reverse, that is also mentioned here. Yeah, and this, if you want to get more information about the gene, then you can simply click on here, and this will take you to the NCBI site where this monosidase beta gene will be present. But if you simply keep your cursor here, then also it will give you the brief information that from which organism this, in which organism this gene is present, what is the name, what is the gene ID in NCBI, how many transcripts are available, how many proteins have been identified, then how many genes it is associated with, and the PubMed. PubMed means how many publications are available for this gene. You can also look into the orthologs of this gene, which means that are there some other organisms also which have this gene. You can also go to Genome Data Viewer and you can also perform its blast. Yeah, so key observations also tells you the same. I've kept it because if you have not attended the lecture, then also from this particular key observation section, you will be able to find out that what is being shown in this figure. Yeah, and then there will be a user guide. So is it clear till now, or do you have any query about how it is working? Okay, you have asked. So is 6-frame translation is specific for prokaryotes and 3-frame for eukaryotes? No, 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 this is not the thing. 6-frame will also work for eukaryotes also. What are the red tiles with star on it? Let me check. Where was the star in the red tile? Means I haven't come across this information that there is some star, but star is actually whenever the star is written somewhere, it means that that thing is conditional. So I'm not sure because I have not come across in IGV that there is some star, but red tiles generally means that it is a stop codon, but if star is written, that it might mean that it is conditional, which means that uh, due to some mutation or some variant has caused that stop codon, or it means it might not remain always the stop codon, it might vary. So star means it's conditional, but I have not seen that. Means I'm just speculating that it might mean that. And about the six frame translation, so it is in both the prokaryotes as well as eukaryotes. Because three frame from the forward strand and three frame from the reverse direction. So in that case, there are six frames which are possible. Yeah, so any further query? I'll also check this question in the assignment as well. If I'll find it, then I'll also check it that what does it mean. And I'll also put it in the end of the presentation when I'll upload my presentation. So I'll check that what does it mean and I'll put it in the end. So you'll be able to find it. Okay, so now we'll see that. Did you ask anything else? Okay. Due to the replication mechanism, will there be any difference? A replication mechanism among prokaryotes and eukaryotes you're talking about? I think replication is same in both. There is no such difference in the mechanism, although different enzymes are used, but in both of them, the replication is bidirectional only. Yeah, both have bidirectional. But one of the main differences is that in prokaryotes, as their DNA is circular, so they have only single origin of replication, which will synthesize the DNA on both the directions. It will be bidirectional replication. And in case of eukaryotes, we have multiple origin of replications, which are known as ARS, autonomously replicating sequence. So in both of prokaryotes and eukaryotes, the directionality of replication is bidirectional only. And replication mechanism is also the same only. So in both of them, if we look in the perspective of frame translations, there are six possible frame translations which will be present in both of them. Yeah. Yeah, so now if you further want to get any information related to IGV, then you can simply go to their user guide in which each step by step they are telling you about each information, how you will be able to explore it. So these all things we have already discussed, that how we can view our results in the IGV. And in this, they have explained it in detail. So I think if there is anything related to the STOPE star, that will also be present here. So these are the sequence tracks. This is telling you about the sequence tracks. And then you can also view them as a colored letters. So because I have increased that scale now, that's why my sequence was visible. But what if I have not scaled it? If I am focusing on entire chromosome or entire genome, then in that case, it's not possible to write these nucleotides. In that case, it will be represented in the form of colored bars. And in case of if I have increased its size, increased the scale, then it will also be shown in the terms of colored letters. This is the DNA sequence. And each color is specific to one particular nucleotide. For example, green for adenine, similarly for blue for cytosine, yellow for guanine, and red for thymine. Then in this, they are telling about the strand direction. So initially, the forward or positive strand is displayed. So in that example, as we have seen, I'll just quickly go to that. Man BA. So this is the positive strand for which the information is shown here. So as you can see, there's stop, stop, stop. So in this gene, we are not able to get the means we are not able to find that whether it is coding. So in that case, they have mentioned that it is this is the coordinates and it is the negative. That means it is starting somewhere from 770 and moving towards moving towards the 452 in the negative strand. So when I will count from the negative direction, then this gene will be identified. So which means that in both the directions, because replication is done in both the directions. So in both the directions, there is a possibility that one particular gene will be synthesizing a protein from negative direction, which will be my uh, uh, reverse direction or the reverse strand. So it will also tell you about the strand direction. So by default, whatever you see in the three frame translation, it will be a positive strand or a forward strand. And you can simply go to the reverse option, which is present on top of 3' translation. So in that case, you will be able to reverse it. And then further, you can also perform the 3' translation of that reverse strand. Then in this case, sequence translation. So they have mentioned that how you can see the sequence translation. You can click on 3' translation, and this is what they have shown, that there are start codon positions and stop codon positions for each frame. Then genome annotation tracks. So in this case, for each gene also they have mentioned that in particular chromosome, what is the position of that genome, position of that gene. That also you can capture here. And navigation, the view. So in this, they are telling about how you can navigate across chromosomes. You can also enter the coordinates, and then you'll be able to see. So these all things we have already covered. Yeah, so if you want to read more in about it in more detail, then you can go to the user guide. So the question which you have asked about these stars, so maybe you will be able to find it in the user guide if that particular thing is available. Now we will discuss few MCQs. So should we start with the MCQs? Yeah, okay. So we have 15 minutes. So the first question is that what was the name of the scientist who distinguished Hodgkin and non-Hodgkin lymphoma based on the presence of distinct cells? Option A, Muller. Option B, Dorothy Reed. Option C, Virchow. And option D, Florence Sabin. So I think this was discussed in the last lectures, which were given by the Dr. Sachin, I guess. Option C, Virchow. Okay, what about others? What do you think? Because here he has specifically mentioned this in his lecture that one particular scientist was the one who distinguished between the Hodgkin and non-Hodgkin, and he has also mentioned that how these two lymphomas were distinguished. Okay, now if nobody else is responding, then let's see. So the correct answer is Dorothy Reed, and he has also mentioned this name. So now we'll also look into the other scientist as well. So when we say Muller, so he was a German pathologist, and he is known for his contribution to physiology. So he's not related to lymphoma classification, and he has done contribution to human physiology. Then Dorothy Reed, and who was the American pathologist, and in 1902 they described the distinct large abnormal cells which are known as Reed-Sternberg cells. And Reed means from his name only, the Reed term is taken. So there are Reed-Sternberg cells, which are the characteristics of the Hodgkin lymphoma. So when these cells are present, so which means they are associated with the Hodgkin lymphoma, and if they are absent, which means it is a non-Hodgkin lymphoma. So Dorothy Reed was the one who was the first scientist who distinguished between the Hodgkin and non-Hodgkin. When we say Virchow, so he was a German pathologist, and he is also known as the father of modern pathology. But he did not classify lymphomas. But his contributions are although significant related to cell theory and many diseases. So I think Rudolf Virchow was his full name. And then Florence Sabin. So I think she was the American medical scientist, and she is also the pioneer woman in science and medicine. But her work is specific to embryology and anatomy. So the answer is Dorothy Reed. And Dorothy Reed was the one who distinguished between the Hodgkin and non-Hodgkin, and by the presence of Reed-Sternberg cells. So I hope now you'll be able to remember this. Now the second question is, yeah. Yeah, sorry. Dorothy Reed is the female scientist. So next question is, when was the first time a definite gene signature was attributed to a cancer? Option A, patients of chronic myeloid leukemia have smaller chromosome. WHO classification of 2008, WHO classification of 2016, and molecular profiling of DLBCL. So you have to tell about the first time when the gene signatures were attributed to cancer. I think this might be confusing. Okay, option D, option C. sorry. Yeah, so the correct option is option A, because they have mentioned it first time. So that's why patients of chronic myeloid leukemia have a smaller chromosome. This was the first signature which was attributed to cancer. So I have discussed each of them, each of these options as well. So the discovery of Philadelphia chromosome, which was made in 1960, was the first identification. And this Philadelphia chromosome was also discussed in that class, in the lecture also, that it is the how the translocation has been done between chromosome 9 and 12. We'll look into the Philadelphia chromosome in the next slide. So this was the first study which was behaving like a gene signature attributing to a cancer, and which was associated specifically with the chronic myelogenous leukemia, which is CML. And then if we talk about WHO classification of 2008, then it provided a refined classification of lymphomas incorporating genetic information, but it was not the first study. Similarly, WHO classification of 2016, it was the updated classification in which more genetic and molecular data was present, but similarly, it was not the first instance. Similarly, molecular profiling of DLBCL, this which was the diffuse large B-cell lymphoma. So it also defined the gene expression signatures for the subtypes, but it was not the earliest instance. So although molecular profiling was also dedicated to gene signatures only, but in this case we are only focusing on the first time. So first time was the patients with the CML have a smaller chromosome. That's why option A was correct. Otherwise, D option could also be correct if only gene signatures would have been asked. Yeah, now we'll see that what does Philadelphia chromosome means. So whenever in chromosome 9 and 22, if there is some translocation of a chromosomal segment, so what can happen is that in 22 chromosome there is a gene which is BCR gene, and in chromosome 9 there is a ABL gene. So whenever this translocation happens, the bigger segment from chromosome 22 breaks off, it gets transferred to chromosome number 9, and a smaller part of chromosome 9 which contains the ABL gene, it gets transferred to the uh, chromosome number 22. So in this case, what happened is chromosome number 22 gets shortened because it has translocated. There is an imbalance translocation. So in that case, chromosome 9 will be enlarged or elongated, and chromosome number 22 will be shortened. So in CML, it was identified, and they have mentioned that it is a signature. Whenever this Philadelphia chromosome is present, we can simply say that it is associated with the chronic myelogenous leukemia. And what it does is, if BCR and ABL will combine, they will form a fusion gene, which will cause immature white blood cells to grow uncontrollably. So whenever immature blood WBCs will grow, they will build up in the bone marrow and blood. So in that way, they leads to CML. Yeah. So in they have also mentioned that some people with acute lymphocytic leukemia or acute myelogenous leukemia can also show this particular signature, but mostly it is associated with the chronic myelogenous leukemia. Yeah, so this was the first signature which was found. Now, next question. So, cancer develops as a result of accumulation of somatic mutation and other genetic alterations that impair cell divisions, checkpoints, etc., which results in abnormal cell proliferation and eventually tumorigenesis. Which among the following statements is not correct regarding the role of mutations in cancer? So option A is genes whose mutation facilitates tumor growth are called driver genes. Driver mutations denotes mutations under positive selection with a population of cells. Option C is along with driver mutations, there may be passenger mutations which does not promote the growth of cancer cells. And option D, all mutations in cancer genomes contribute to malignant initiation of progression. So now you have to select the option which is not correct with perspective of role of mutation in cancer. Okay, so Polemy has mentioned option D. Anyone else wants to guess any option? Okay, so we will see. So yeah, the correct answer is that all the mutation in cancer genomes contribute to malignant initiation or progression. So although there are multiple mutations which might be observed in cancer, but that doesn't mean that all those mutations are means contributing to the malignant nature of cancer. There are some mutations which might be a byproduct, because in cancer some genes will be affected, some sequence will might have gone under, means undergone change. So due to that change, everything is impaired. So in that case, means in replication also, as we know, the DNA polymerase has also some kind of, means if we talk about its fidelity, so it's not that accurate. It can make some mistakes. So with mutation, replication and every other mechanism is also impaired. So in that case, other mutations can also arise as a byproduct of my main mutations. So in that case, not all the mutations contribute to malignant initiation. Some mutations just are like a passenger mutations. So we'll see them. Okay. So in this, I've not added that. So when we say driver mutation or driver genes, those genes are the ones which are helping the tumor growth. So these genes, if there is any mutation in these genes, so they will be associated with the tumor growth. Whereas the driver mutations denote the mutations under the positive selection with the population of cells. So in this case, it means that driver mutations are the mutations which are under positive selection. So those mutations, if they are occurring, they will lead to the cancer phenotype. Whereas if those mutations which do not contribute anything to cancer cell, they are referred to as passenger mutations. They are acting like a passenger. They have no role to play. If they occur, then also no impact on cancer. If they do not occur, then also no role in cancer. So if we perform gene therapy, or if we try to take those genes as a biomarker, then it will not help us. So that's why it is very important to identify that which mutation is a driver mutation and which is a passenger mutation, so that we can design better therapeutics which can target. So that's why there is a term called blind therapy. So that is one of the reasons that we are not able to stratify people, and those we are not able to classify mutations, that which mutations are driver and which are passenger. Now next question is that passenger genes are not ultimately pathogenic, but mapping them is also important. So which among the following reasons describe the importance of passenger mutations? So option A is they lead to discovery of driver mutations. Option B, they expose the DNA repair pathways imperative to cancer development. They shine light on the previously incurred DNA damage. And option D, they work as a unique signatures for a particular tumor. So you have to tell that which among them is the reason that why we should also focus on the passenger mutations. So these are the four options which are taking these points. Okay, Polemy has mentioned D. D means this four, right? All the A, B, C, D, or you are simply meaning D? All, okay. So what about others? Anyone else wants to guess anything? Okay, now let's see. So the correct option is A, B, C, D. Because the passenger genes, although their mutation is not contributing, but they can also lead to the discovery of driver mutations. They can help us to discover the driver mutations, and they also expose the DNA repair pathways, which are imperative to cancer development. And they also shine or put some light on the previously incurred DNA damage. So we can perform kind of comparisons we can perform. If we know the passenger mutations, then we'll be able to figure out, we will be able to cancel out passenger mutations, and we'll be able to capture the driver mutations. And they will also work as a unique signatures in particular tumors. So there are some tumors which has some driver mutations, and then there are some passenger mutations which might be specific to those tumors. So in that case, those identifications will also be helpful. So that's why they can also act as the unique signatures. So in this way, all the options are correct. Now the next question is, which of the following is or are correct for the affinity-based proteomics? So option A, it is the field of proteome analysis based on the use of antibodies. Option B, it is the field of proteome analysis based on the use of antibiotics. Option C, antibodies and other binding reagents used as protein-specific detection probes. And option D, it can be used to enhance the biomarker discovery and validation. So in this case, more than one option can also be correct, and you have to tell that which among them is correct for the affinity-based proteomics. Okay, CD and ACD. Okay, now let's see. So yeah, ACD are correct. because it's not it's not using anywhere. Antibiotics are not related to the affinity based, but it's more dedicated to antibiotic based. So yeah, in this proteome analysis is based on the use of antibodies and not antibiotics. Then antibodies and other binding reagents, they are used as protein specific detection probes. So if you want to identify any specific protein, so you have to design the antibody which is complementary or specific to that protein so that you can identify that particular protein in your sample. And then it also enhances the biomarker discovery and validation. So yeah, this is also true. Yeah, so this was the last question. That's it for this session. So do you have any further doubts before ending the session, or do you need any study material or anything? Okay, thank you. Okay, thank you so much. So I will end this session here. And now in the next session, we'll be talking about the next week. And in this assignment, I think there were no such questions which were very confusing. So this week assignment was quite easy. So you'll be able to solve that. And for the previous, I'll go through the discussion forums and I'll write. So thank you so much. We'll end this session here only. See you next week.